Gram staining. Welcome to microbiology class. Why do we do gram staining? Bacteria are transparent and must be stained to be viewed under a microscope. Gram staining is one of the simplest and most practical bacterial staining methods. The basis of gram staining is the difference in the structure of bacterial cell wall. Based on their cell wall characteristics, bacteria are classified into 5 main groups, in cloud, gram-positive, gram-negative, acid-fast, wall-less, and archaebacteria. Gram staining identifies and visualizes the 2 largest groups, gram-positive and gram-negative bacteria, which together account for about 95% of the global bacterial population. The remaining 3 groups, representing roughly 5% of bacteria, require their own specific staining methods. What happens in gram staining? Gram-positive bacteria possess a thick peptidoglycan layer in their cell wall, which enables them to retain the crystal violet iodine complex and resist decolorization during the acid-alcohol step, thereby remaining purple. Contrast, gram-negative bacteria have a thin peptidoglycan layer and an outer lipid membrane. During the acid-alcohol destaining step, their outer membrane is disrupted, allowing the original dye to be removed. Therefore, they require counterstaining to appear. Materials: clean slide, sterile loop, Bunsen burner, water, microscope, staining reagents. Primary stain, crystal violet. Mordant, Lugol's iodine. Destainer, alcohol or acetone. Secondary stain, safranin or fuchsin. The procedure. Performing a gram stain involves 5 main steps, each of which is essential. Step 1, preparing and fixing the smear. Step 2, staining with crystal violet. Step 3, fixing with Lugol's iodine. Step 4, destaining with alcohol. Step 5, counterstaining with safranin. Step 1, preparing and fixing the smear. A good smear is a thin and even layer of bacteria on the slide that sticks well and does not wash off during the staining process. Smear preparation consists of 3 main steps. First, making a smear from the bacterial culture. Then, air drying. And finally, heat fixation. Of course, the way smears are prepared from liquid and solid media is a little different. To prepare a smear. From a broth culture. Use a sterile loop to place a drop of the culture in the center of a clean slide. Then use the loop to spread the drop on the slide. But to prepare a smear from a solid culture, first, place a small drop of distilled water or saline in the center of the slide. Then, using a sterile loop, pick up a tiny amount of a bacterial colony and mix it into the drop. Spread it gently in a circular motion to form a thin, even layer about 1 to 2 centimeters in diameter. Then allow the slide to air dry completely at room temperature. This process may take a few minutes. After the slide dries, hold it with a forceps and quickly pass it through the Bunsen flame 3 to 4 times. Remember, do not overheat it. If it gets too hot to touch, the bacteria may be damaged. Step 2, staining with crystal violet. Now your slide is ready for staining. Place the slide on a staining tray. Cover it with crystal violet. After 60 seconds, at this stage, all bacteria appear purple. Then, rinse the slide gently with water to wash off extra dye. Step 3, fixing with Lugol's iodine. Cover the slide with iodine for 60 seconds. Iodine acts as a mordant, forming a large crystal violet iodine complex. Rinse the slide gently with water to wash off extra dye. At end of this stage, all bacteria still are purple. Step 4, destaining with alcohol. This is the most important and critical step. Add a few drops of alcohol for 10 to 20 seconds. Gram-positive bacteria stay purple, but gram-negative bacteria lose the color and become colorless. After this step, rinse the slide immediately with water. The last step is counterstaining with safranin. Cover the slide with safranin for 60 seconds. Gram-negative bacteria turn pink or red, while gram-positive bacteria stay purple. Finally, rinse the slide with water and dry gently. Interpreting the results. Now the bacteria can be observed under the microscope. Gram-positive bacteria appear purple or dark blue, and gram-negative bacteria appear pink or red. Quick tips. Key factors in gram staining. 1. Smear preparation. The smear should be thin and uniform, using only a small amount of bacteria. Thick smears can interfere with staining and decolorization, leading to false results. 2. Heat fixation. The slide must be completely air-dried before heat fixation, otherwise, residual moisture may boil and damage the cells. The slide should be passed gently through the flame, as overheating can kill or distort the bacteria. 3. Age of culture. Young cultures, 18 to 24H, are recommended. Older gram-positive cells may lose cell wall integrity and appear gram-negative. 4. Decolorization. Decolorization is the most critical step. Insufficient time may leave gram-negative cells purple, false positive, while excessive time may decolorize gram-positive cells, false negative. The typical duration is 10 to 20 seconds. Finally, proper gram staining provides rapid and essential information for preliminary diagnosis and helps guide appropriate antibiotic therapy.